So, as I have told you that there are two phases of biotransformation reaction. In the previous session, we have discussed about the phase 1. Now we are going to discuss about the phase 2 biotransformation reaction. Here, there is in the phase 2 biotransformation reaction, the most important is the drug or its metabolite. It combines with the endogenous substrate which is derived from the carbohydrate or lipid metabolism, and that's why it is also known as a synthetic reaction. It is also known as a synthetic reaction. And as the combination is there, the molecular size is increased and the compounds become more polar plus larger, so it is ultimately excreted from the body. Now, which are the different phase 2 biotransformation reaction? The most common, you have to remember like this: in the phase 1, the most common is the oxidation, while in the phase 2, the most common is glucuronidation. And this glucuronidation reaction, it is the most common and it occurs with the help of the group of the enzymes that is known as a UGT, and that is UDP glucuronyl transferases. Okay, and the most common example is oral contraceptive pills. Okay, and the another example, if you want to remember, that is chloramphenicol. Now the one point or one question may arise in your mind that, sir, you in the previous session you have told that this chloramphenicol is metabolized by the reduction. So here I want to tell that there are more than one, there may be two, there may be three different biotransformation reactions by which a drug can metabolize. Okay. Now, even certain endogenous substances like bilirubin, it is also metabolized through this glucuronide conjugation. Okay, and we have discussed this point categorically at the time of enterohepatic recycling. At the time of enterohepatic recycling, that the drug that is going to be conjugated, drug plus conjugate. Now these drug plus conjugate, they are excreted in the bile, reaches the intestine. Again in the intestine they are split off by the bacteria, and again the drug becomes free, and this drug is again reabsorbed and then again metabolized. Now cycle, this cycle goes on and on. So you may think that why I am repeating this right now, because the most important point to gain the knowledge and to become expertise in the medical field is you should try to correlate the different points. Okay, that is one thing, and the second thing is that you should have a broader perspective or the broader vision and the holistic view. Okay, so here I want to add one more point in addition to this enterohepatic circulation, that I have told you that at the end of phase 2 biotransformation reaction, the majority of the drugs, it becomes inactive. But one of the principal mechanism by which the morphine is metabolized is through the glucuronidation, and this morphine is converted into morphine 6 glucuronide. Okay, and these metabolites is active. So this is all about the glucuronidation reaction. Now there are different type of the conjugation reactions. The second is the sulfate conjugation. Then there is a glycine conjugation. But these are not that much clinically important. Okay. Now the next that you should remember is the acetylation. Acetylation. Acetylation of the drug occurs with the help of the acetyl coenzyme A, and there is a significant, here that you should remember is there is a significant genetic polymorphism, and that's why there is a different status of individual for the metabolizing enzyme. Say for example the acetylator status of the individual. Some may be slow acetylators, some may be fast acetylators. Now as we have discussed that except few cases, at the end of the phase 2 reaction, the drug becomes inactive. So, if the person is fast acetylator, if the person is fast acetylator, then what will happen that quickly the drug becomes inactive. So you have to frequently administer the drug, which is not required in case of slow acetylators. Okay, and the classical example is isoniazide. Classical example is isoniazide. It is metabolized through the acetylation, and there are certain slow acetylators and certain fast acetylators. In slow acetylators, if you can give the drug on the alternate day regime, then still the plasma concentration is sufficient, which is required for the treatment of tuberculosis. But the same is not possible in case of fast acetylators. Okay, but now the question is not there in the case of the tuberculosis, because as you know the revised guideline, this isoniazide is to be given daily, but you should know this. Okay, now one we have covered the conjugation, second the acetylation, then the next is methylation, conjugation. Now as such these reactions are not as such these reactions are not that much important, but in case of glutathione conjugation you must remember one point, that after this glutathione conjugation, say for example the paracetamol, when the paracetamol is metabolized, the reactive intermediates like the epoxy metabolites, these are produced, and these are very short-lived but toxic intermediates, and these require this glutathione conjugation to make this toxic into the non-toxic metabolite. Now suppose you have given the higher dose of the paracetamol. Ultimately what will happen, that there is a depletion of this glutathione, and that's why there is a not conversion or the removal of these reactive metabolite, and it will lead to the toxicity. Okay, and the last phase 2 reaction is the ribonucleotide or nucleoside synthesis. Okay, ribonucleotide or ribonuclear synthesis. Now this is mainly important in case of, you all know that there are various anti-metabolites. These are used, purine anti-metabolites, pyridine anti-metabolites, these are used in the cancer chemotherapy, and this pathway is important for the activation of these anti-metabolites. Okay, so in summary, at the end of phase 2 biotransformation reaction, the ultimately the drug or its metabolites become highly water soluble, larger molecular size, definitely eliminated, and the most important phase 2 biotransformation reaction is the conjugation. Again, in the conjugation there are different types, but the glucuronide conjugation is most common. Okay, then there are glycine conjugation, sulfate conjugation, glucuronide conjugation, methylation, and ribonucleotide synthesis. So this is all about phase 1 and phase 2 biotransformation reactions. In the next session, I will talk you about the microsomal enzyme induction and inhibition. Thank you.